So I think these initiatives say something about, his drive in doing science and his motivation. And in the past few months that I was away, my lab relocated to Yale. Gian Marco was adopted by William DeWitt here in Genome Sciences, and they did also some beautiful work here together. And so speaking of that, he also speaks of the power, so to speak. So there was a recent paper out recently by ARC Institute. For some of you, it's an interesting institute, let's put it that way. Claiming things they weren't quite proving right. And Will and Gian Marco together, they wrote this commentary that basically went to the belly of the beast. And it got a lot of traction online. People basically acknowledging the fact that, you do good science, you need to do good assessment of the model you have and show, the claims that you're putting out there, they need to be warranted. And these two actually show that those claims were not warranted. And I think it's a very nice work. One thing I didn't put in the slides, I just saw William today, I should mention that. So William was an undergraduate student from computer science in our lab, recruited by Gian Marco. And Gian Marco has been really involved in training undergraduates and graduate students in the lab and doing an amazing job. He's a way better mentor than I am. And so there's a lot to learn there. So that's something to say. But it's not all about science, right? Adventures continue. So I think if you look at some of his, the way he would describe himself is like, I think the first is he's an aspiring chef or a chef, and then a scuba diver, and if he has time, he does some science. So I don't know if that's true or not, but yeah, so there's a scuba diving adventures, mainly with Kevin here, who also has the same passion. And then, we, on the sort of Stand Up for Science Day, our lab had a afternoon retreat in the Snoqualmie Pass skiing, and so Gian Marco printed all this science, so we skied around with the science behind our back. So I hope you get a sense of who is going to give this presentation today, and what kind of person he's been, and the work that he's been doing in the past few years. It's been really a pleasure having him as lab and sort of learn from him and work with him. And I hope you see some of that work today. You appreciate it as much. Right?
Thank you.
Thank you.
All right, thank you, Armita. That was not that much of a roast. I think you were nice for the most part. All right, so thank you everyone for being here. Welcome to my thesis defense. Thank you for my committee in particular for taking the time to evaluate me. To Armita, Bill Noble, Chris on Zoom. Thank you for the last minute addition, and Alex as well. So today I'm going to talk you through most of the work that I've done for my PhD, which essentially revolves around modeling and designing proteins with the help of these rotationally equivariant neural networks that I will talk about. So since this is a computer science thesis, we're going to start by introducing proteins. So everyone here likely has heard about proteins one way or another. Perhaps you've heard about specific type of proteins in high school, like antibodies or ion channels, without maybe knowing that they were examples of proteins. Because in fact, proteins are an extremely diverse class of molecules that carry the majority of the functional roles that underlie life. And this great functional diversity is granted by an immense diversity in how proteins can be composed. Each protein is composed of a unique sequence of small molecule residues that can have 20 possible types called amino acids, shown in the bottom left. that come in different sort of shapes, sizes, and physical-chemical properties. And to form a protein sequence, the residues are chained together with covalent bonds along a conserved region of the residues that is called the backbone, and together they form the backbone chain of the protein, and instead the variable part of the residues, which identifies them as a specific amino acid type, hangs off the side of the chain, and so it's called the side chain. And the specific amino acid sequence of the protein, which gives it different biochemical properties, endows the protein, every protein, with the ability to engage in very specific interactions with other molecules in its environment, whether these are other types of proteins or ions, small molecules, DNA, or what have you. And it is the nature of these interactions and the way they're carried that then define the function of the protein. And the reality of how these functions, these interactions happen in nature is very dynamic and messy and quite hard to probe in its entirety. But luckily, to a great degree, or in most cases, this can be simplified by noting that the way in which functions are carried by a protein is often mediated by the specific 3D structure that the protein takes. In fact, most proteins tend to fold into one or at most a few stable 3D structures, which then largely determine the types of interactions that the protein can carry. Like, you can think very naively about an ion channel whose job is to pump ions through membranes, and as the name implies, in order to carry such function, it has to adopt a certain shape. This is a very simple example, but likewise, all interactions are dictated by, many interactions are dictated by the shape of the protein. And so within this slightly simplified view, there are then two physical quantities that I will talk about at some point in the presentation that are often used to characterize a protein's behavior and the way it carries the function, and they can also be experimentally measured. So one of these quantities is the stability of a protein structure, which you can think of as the propensity of the protein to stay in a particular structural fold. And this is quantified by this value shown here, delta G, which is the difference in what's called the Gibbs free energy between the folded and unfolded states of the protein. And we can also use a very similar quantity, which is also different in Gibbs free energies, to instead measure what's called the binding affinity between two proteins or two molecules in general, which you can think of instead as the propensity of the two molecules to want to interact, to want to stay bound together, which again is at the basis of many of the functional roles in proteins. And very often in scientific discovery, as well as in engineering applications, as we will see, we're actually most interested in how these two quantities change as a result of mutations or variations in the amino acid sequence, and we usually quantify these with these delta G values. And again, there do exist wet lab experiments that measure these quantities, and even experiments that resolve the protein structures and the structure of protein-protein interactions. But these experiments are very expensive, laborious, and require a lot of expertise. And so this brings up the desire to instead want to model proteins and make predictions about proteins from a theoretical and computational standpoint, so as to minimize the need for wet lab experiments, or sort of reserve them for sort of the last validation, and to do it sort of less often and more cheaply. And there are several popular approaches to model proteins, and I will just walk you through them in a way that serves my thesis. So One approach is called molecular dynamics, which essentially attempts to explicitly model the dynamics of protein folding and protein binding by simulating through time the results of the fundamental interatomic forces that are at play in folding and binding. And this approach is often cited as being very accurate, with some shortcomings. But the biggest shortcoming is that it is extremely slow and requires very specialized hardware to run. So it's actually not practical to try and understand proteins using molecular dynamics for most applications. But it's instead a more practical alternative that still does, however, attempt to model the fundamental physical interactions from first principles, is using these energy scoring functions such as Rosetta, which are essentially handcrafted functions that score protein structures and protein-protein complexes in such a way as to make the experimentally measured true structures live in low energy wells of the predicted energy landscape. And so because of this, people use these energy functions to guide the design of proteins and protein-protein interactions, so such as to go into low energy wells of the landscape, which corresponds to, again, stable structures. And these, because they're much faster than molecular dynamics, these are very broadly used by people in the field, but they're not always accurate, and they're still often too slow for certain high throughput applications. And so finally, what I'm going to focus on today is this third approach, which we do take in this thesis, and which the whole world is basically turning to, which is to mostly ditch first principle thinking and just train machine learning models. And so in machine learning, instead of relying on explicit physical laws to then make predictions about proteins that we may care about, like you do in the first two approaches, we instead try to learn directly from data how to make the predictions that we care about. You can think of it as trying to learn the first principles from data. And specifically what we do is we train these complex models, usually neural network models, on some training data that hopefully captures the relationship between parts of the proteins that we care to model and predict. And we have the important goal of having these models be able to make accurate predictions beyond the data that was used to training them, which in machine learning we call the ability to generalize across the sort of physical landscape. And in my view, having this type of generalizability simplified generalizability is akin to learning what physicists may call an effective theory, and to learn this from data. So in physics, an effective theory is a model, like a mathematical description of a process that purposefully ignores or integrates out some known details of the process in such a way to ignores details that are either not important or that you can simply afford to ignore for the specific application that you care about. And again, I think that combining this simplification, but making sure that it generalizes across the physics is what I think machine learning models can learn, and what ideally we want them to learn, because they would provide us with predictive tools that are both very, very fast, but also have generalizable accuracy. And when developing machine learning models, as you can see here, we have a lot of freedom of choice in the types of predictions that we can train for, and even in exactly the sort of how to frame the prediction problems we care about, like exactly what aspects of the data to include in the prediction. And it's important to make these decisions very carefully, because every choice may result in learning a different effective theory, and some theories may be more generalizable than others. And a good rule of thumb in this design process is that the closer the modeling choices you make are to the physical reality that you want to capture, then the better the chances you have at capturing such physical reality in the models that you train, instead of potentially having your model learn some shortcuts that do not generalize. And so with this idea in mind, in this thesis, I've primarily focused on modeling protein structures, and whenever possible, the full atom representation of the structures, since we know that these two elements are very close causal drivers of what drives a protein function. And in particular, I have focused on building neural network models of local atomic environments that surround individual residues in protein structures. So there's two elements of this, right? So the choice of focusing only on modeling individual residues is because residues are natural building blocks of proteins, as I've discussed earlier, and many things that we care to sort of model and predict about proteins, such as the effect of mutations, as we will see later, can be decomposed at the residue level. And then instead, the choice of only making the predictions depend on the local atomic environment, the local environment, is driven by the consideration that since the laws of physics are applied locally, then we can assume that any physical property that we care to predict that is driven by the residue or some change that is happening to the residue is going to be dependent mostly upon local interactions that are occurring between the residue and its immediate environment. This is just an approximation. It's not always, that's not always true, but sort of simplifying the problem this way also simplifies greatly the modeling, which means that we're actually more likely to learn a generalized model, a generalizable model that generalizes within the sort of limits of the local domain that we're training it for. Because instead, if we were trying to model the whole protein, and once we're actually at more risk of we say machine learning overfitting, which means not generalizing essentially. And finally, as a last element of this framework, another thing that the laws of physics tell us is that any property that, any physical property that we will care to predict about a residue is going to have some sort of rotational symmetry with respect to the input residue, meaning all the properties are either going to be invariant, aka unchanged, as a result of rotations of the input, or they are going to rotate alongside it, in which case we call them equivariant. And to make sure that every model that we learn about neighborhoods, it also possesses these properties, we constrain our neural networks to be rotationally equivariant, meaning to have these rotational symmetry baked into the predictions that the model can make. And I won't go into the details of how exactly these neural networks are built. I'll just highlight a couple of the most important elements, which is that first, in order to construct these neural networks, we have to encode these atomic neighborhoods into a convenient representation that then allows us to build the neural networks. And specifically, this representation is like a Fourier transform onto a basis of spherical harmonic functions and regular basis functions, where the spherical harmonics component is the crucial part, because spherical harmonics have certain properties that allow them to play really nicely with rotations. And in fact, once the atomic point cloud is encoded into a very special basis, then there are rules that mathematicians and physicists before us have worked out to build mathematical functions, and therefore also neural networks that respect rotational symmetry. So fundamentally, building these neural networks is just respecting these rules that someone else has laid out. And so throughout my PhD, I have used and refined this equivariant framework, and with it I've developed 3 different models, which I will now take the time to discuss. Starting from the first one I worked on, which as Amrita mentioned is the one that was the most theoretical, where I just really tried to learn the rotational equivariance. And this model essentially was a rotationally equivariant autoencoder, which is a neural network that is trained to reconstruct the Fourier encoding of atomic neighborhoods, again in an equivariant way. So if you were to rotate the input, the reconstruction would also rotate in the same way, which makes the reconstruction laws work very nicely. And in the process of learning to make this reconstruction, the neural network learns a information-rich embedding that represents the neighborhood in a rotation invariant way, and that is also coupled with a rotation matrix shown there in red that instead sort of captures the equivariant part of the neighborhood, so the orientation of it in 3D space. And we found that these invariant embeddings capture key structural properties of the neighborhoods. For example, here I'm showing a UMAP projection of the learned neighborhoods that captures the dominant secondary structure element of each neighborhood. You can think of a secondary structure as a local structural motif in the protein structures that is important in the modeling and understanding of the structures. But most importantly, we find that these embeddings can serve as effective representations and information-dense representations of the local structure environments for downstream machine learning tasks. And so as a proof of concept, what we did is we used our autoencoder to extract embeddings from the binding pocket, the structural binding pocket of proteins, small molecule ligand structures, and with them we constructed numerical representations of these structures that we then used as the representations for supervised machine learning models to predict the binding affinity between the protein and the small molecule. And we found that with this very simple approach, we were able to achieve state-of-the-art results in predicting binding affinity. And so these results then prompted other work that is still ongoing in my lab to try to use these embeddings as representations that can help relate structural features of proteins with the specific functional family that a protein belongs to, like asking why does this protein carry this function? What about the structure makes the protein carry this function? Moving on then to the second project, which instead I worked on, sort of switching gears completely in many ways. I worked on a task called sidechain packing, which is essentially the problem of figuring out the precise atomic conformations of the residue side chains that are consistent with a particular backbone representation of the protein structure. So just the backbone attached together without a notion of the side chains, and with a amino acid sequence that is predicted to fold into the backbone structure. And so the sidechain packing is one of the standard sort of sub-tasks in many protein design workflows, because many people tend to first design a backbone, then figure out the sequence, and then they have to get the full atom structure with some other tool. And so this is the project that, as Amrita mentioned, I started working on with William, a brilliant undergrad who now works at Xera, also still on proteins, and I like to think that he's there because of the work he did here, so I'm happy for him. And so together, after, he worked on it for me for a bit, and then he moved on to better things, and so then I carried the project to completion. But essentially we developed this tool called HPacker that essentially predicts sidechain conformations through predicting the dihedral angles, specifically called chi-angles in this case, that describe univocally a sidechain conformation, and that are, of course, invariant to rotations. And specifically, our method predicts the angles for each residue in parallel across the entire protein using local models. And first, we use a model of backbone neighborhoods together with the amino acid labels to make an initial guess for the chi-angles and therefore the conformations. And then once we populate the structure with these predicted initial conformations, we found that if we use a all-atom model, of the neighborhood, so a model that was trained to predict sidechain conformations from the true all-atom configurations, applying this iteratively to the predicted structure actually further improved the performance. We think because it allows the prediction to depend more specifically on the atomic interactions that are occurring. And in the end, so when we sort of tested these model on common benchmarks, it seems that the model is on par with most of the state-of-the-art tools for predicting sidechain conformations, and it's also pretty fast. But it didn't seem to shine as sort of the best model, so we just submitted it to a conference. But still, that being said, it's the code that we wrote is very broadly used by people in the community. So far, it's actually the most popular thing that I've made so far, for some reason, and which I think it's testament to its effectiveness that maybe goes beyond what these benchmarks can show. And finally, the third model that I will talk about and that I sort of devoted a bit more time to is called Hermes. So I should first note that Hermes is a rehashing of a model that was previously developed in my lab by my former colleague and mentor, Mike Poon, which I re-implemented and improved upon in several ways. And so at its core, Hermes is similar to what people here call an inverse folding model, so a model that tries to predict the sequence, the amino acid sequence, in a manner consistent with a particular structure. So specifically, the model takes as input a all-atom neighborhood of a stable protein structure where the atoms of the central residue have been removed, thereby masking its amino acid identity. And then the Hermes neural network is trained with classification to predict the amino acid identity of the masked residue. And in the process of doing this, Hermes effectively learns the probability that each of the 20 amino acids may fit inside of the input neighborhood. Where importantly, again, these neighborhoods are assumed to come from stable protein structures because that's how Hermes was trained. And therefore, this means that if say we have Hermes predicting that a mutant amino acid has a higher probability of being observed inside of the input neighborhood than a higher probability than the wild type amino acid, we hypothesize this prediction to mean that at least Hermes thinks that the mutant would render the environment more stable with the mutant than it was with the wild type. And so this is sort of the fundamental interpretation of how to use this model, because this means that we can use Hermes as an estimator of the effects of mutations on a protein's thermodynamic stability. However, if we stare at this equation for a bit, which we use to estimate stability effects, we can notice that there's potentially a problem with this term, which is the probability of the mutant amino acid. And specifically, we think this probability is very likely underestimated for many of the use cases we care about, because it is conditioned on a neighborhood that was optimized in the structural sense to host the wild type amino acid. But of course, we know that in nature, we could have cases where there is some mutant amino acid that would fit inside of this neighborhood and even maybe make the protein structure more stable and mostly the same, at least in a way that would keep its function the same, but only if we allow some structural flexibility into the neighborhood. And so our sort of attempt at a solution for this is to incorporate some structural flexibility into the predictions by first relaxing the neighborhood, the structural environment, using the Rosetta scoring function so as to optimally fit the mutant amino acid before then using Hermes to predict the probability of the mutant. And so this in the end gives us two ways of using Hermes at inference time to estimate the effects of stability, of mutations on stability, which we respectively call Hermes fixed and Hermes relaxed. And as I discussed, the relaxed model is more realistic as it's probably able to recover more of the stabilizing mutations because of its ability to consider the possibility of relaxation. And indeed, in a standard benchmark for predicting the effect of stability of mutations on stability, we do observe that Hermes relax has a higher recall metric, which corresponds to an increase in the ability to recognize truly stabilizing mutations. The fundamental problem of Hermes relax, though, is that it is it's much slower than Hermes fixed, like orders of magnitude slower, because you have to run a relaxation for every mutation that you want to score. Like you go in the order of, I don't know, milliseconds to the order of minutes, maybe 30 seconds to make a prediction, which for large-scale analysis is not ideal. And so to try and get the best of both worlds, we came up with a third model that we call Hermes amortized, which is essentially just a fine-tuning of Hermes, such that if you use it in the way of Hermes fixed, it makes predictions similar to Hermes relaxed. We just fine-tuned the architecture so as to make predictions like Hermes relaxed on some subset of the data. And empirically, we find that this model makes predictions as fast as Hermes fixed, but the prediction performance is closer to that of Hermes relaxed, which is quite nice. But what I think is most interesting is to try and understand how the predictions between these models differ the most, to try and understand how to use them and why. And so, in particular, what we found is that the predictions of these models differ most significantly in how they treat mutations between amino acids that have very different sizes. I think this also makes sense intuitively because, again, sort of Hermes fixed makes predictions with this fixed atomic environment, which means that it tends to prefer mutations that preserve the size of the amino acid. Like it's not going to assign high probability to something that is very large or very small, at least on average. And so this means that Hermes fixed has a very hard time recognizing mutations in cases where the mutant differs a lot in size from the wild type, in particular when the mutant is larger, as you can see, whereas the other two models have a much easier time at recognizing these mutations. And finally, Hermes can also be trained directly to predict stability effects. This can be done very simply by just training the neural network with regression by making the predicted difference in log probability corresponding to particular amino acids regress over the experimentally measured difference in stability as a result of that mutation. And so what we did is if you were to train Hermes on the largest available data set of experimental stability effects, which specifically contains experimental stability effects on small monomeric proteins, which is far from being exhaustive of protein space. You get a model that is qualitatively very different from the model that is just pre-trained with classification. Almost all metrics get lower, except for recall, which gets much higher. But if we try to combine the strategies and first pre-train the network with classification and then fine-tune it with regressing over experimental stability effects, we get sort of improvements in performance almost across the board, at least on these specific benchmark, which again is also about on effects on small proteins. And what's nice is that the performance of Hermes, when it is fine-tuned on the same experimental data, it always matches the performance of the best model found in the literature that is also fine-tuned on the same data. Which I find particularly interesting because many of these models have different inductive biases. Like Hermes, as I discussed, is local to the particular parameterized with only the local atomic environment of where the mutation is happening, whereas the model shown here in brown actually sees the global protein, but I think that their matching of result probably implies that it's not necessary, at least in this setting, to model the full protein. But also, trying to optimize these benchmarks is, it's fun to try and do better. It's like kind of like a leaderboard. That's what people in machine learning do all the time. But it's not always indicative of how Hermes can be used to actually help scientific discovery. And again, because these results are specific to this benchmark on small proteins, perhaps the models would perform differently on different domains. And so to try and see if Hermes can be useful in practice, I started a collaboration with Zach Jones here from the King Lab at the Institute for Protein Design, where we looked at the specific application he works on, which is to help design antigens for antiviral vaccines. So just to give you a brief introduction to the problem, which I find absolutely fascinating. So many viruses, specifically envelope viruses, infect human cells by using what's called a fusion protein that is expressed on the surface of the virus, on the membrane. And they use this protein, as you can see here, to latch onto the human cell and fuse the two membranes together so that the virus can inject its payload into the human cell. And importantly, in doing this, the fusion protein changes conformation, going from what's called the pre-fusion state to the post-fusion state. And when making a vaccine, of course we want our immune response, so the resulting antibodies, to intercept the virus before it infects our cells. And so this means that we want the antibodies to attach to epitopes that are presented on the pre-fusion conformation of the protein. But the fundamental problem is that in the wild, the pre-fusion conformation is always less stable than the post-fusion conformation. And so if you were to just make a vaccine using the wild type version of the protein, you would end up with mostly post-fusion conformations in your bloodstream, which would result in a poor immune response. And so the solution that folks in this field have come up with is to engineer the fusion protein that you ultimately put in the vaccine by adding a few mutations, like as little as possible, to preferentially stabilize the protein in the pre-fusion conformation. But so far, the vaccine design community has been lacking efficient computational methods to suggest such mutations. Like, it's unclear which methods work well. In fact, Zach tells me that most people to do this tend to just look at the structure and sort of guess and check which mutations work, and there's sort of limited use of computational tools. And so the first thing Zach and I set out to do was to try to create a benchmark, since no sort of standardized benchmark exists here, to try and test whether Hermes and other models would have identified some of the known pre-fusion stabilizing mutations that we could find in the literature. And I guess I won't go into the details of the analysis, but we just essentially tried to see whether these models would rank the mutants like higher than the wild type and towards the top of the list of 20 amino acids. And in this analysis, we found evidence that the two best performing models at identifying at least this set of pre-fusion stabilizing mutations would have been Rosetta, the energy scoring functions, and to our surprise, Hermes amortized, which is, again, the model that is not fine-tuned on real experimental effects. But the main caveat here is that Rosetta is orders of magnitude slower than Hermes, and it is in fact so slow that it's not possible, unless you have like stupid amounts of compute that not even Zach has in the IPD, to use Rosetta to screen every possible mutation in a fusion protein. And so we thought to try and sort of leverage the strengths of the two methods and propose design pipelines that first score and rank all mutations using Hermes, and then we just take the sort of the top scoring mutations by Hermes and further filter them using Rosetta. So we just limit the use of Rosetta to a handful of high scoring proteins, and then this then gives us the final set of proposed mutations. So as shown here, we actually developed two pipelines. The one shown at the top only focuses on trying to stabilize the pre-fusion conformation by focusing also on mutations in the core of the protein, which usually affects stability the most. And then the second one shown at the bottom is that tries to jointly stabilize pre-fusion while also destabilizing post-fusion. Because again, what matters is just the relative stability between the two. And we were not able to, we haven't gotten around, I guess, to test these mutations yet, because this is very recent work. But we do think that there are some mutations here that are particularly promising, and I just want to highlight a couple because I think they're very emblematic of the type of mutations that we're looking for. So this mutation here, which is A298I, was found by the first pipeline, and it is a canonical example of what people in this field call a cavity filling mutation. And the way this works, as the name implies, this mutation substitutes a small amino acid for a much larger hydrophobic amino acid in a region in the core of the protein, the pre-fusion protein, that is currently underpacked. Like there's just a bunch of empty space, which we know to be not preferred in protein structures, and we know that if you fill empty space, you would stabilize the structure, and this is exactly what we believe this mutation would do. And we further believe this would work because in the post-fusion, this mutation will be at the surface, so it wouldn't stabilize the protein. And I did all this before I actually found out, or Zach found out, that this mutation is actually part of a patent we found later, so it actually does work. We just didn't know until a couple days ago. But I wanted to, give it to you as if we found it. And then the second mutation, which we did not find evidence for later, is also interesting because it's very different in the way it works. So this uses a mutation to a proline. And prolines are very unique among the amino acids because, as you can see here, they can only adopt certain restricted set of structural conformations, and so they're only found. They can only be found in certain parts of protein structures. And one such part in which they can be found and are often found is in like the kinks, like the sharp turns on the loops of protein structures because they make those loops more stable. And in fact, Hermes wants to put a proline in this loop in prefusion. But for the same reason, prolines are also basically impossible to find in alpha helices. Again, simply because of the conformational restriction. And this mutation would be in alpha helix in post-fusion, and so sort of because of this combination of the two, we think these mutations would be quite successful. And so sort of these results and sort of benchmark results from before, I think, highlight that Hermes can be used to very quickly and mostly effectively estimate the effects of amino acid mutations on the stability of the protein, at least in a manner that is useful enough to provide sort of an initial candidate set of things of mutations that can be then further screened by experimental means. But beyond predicting stability, Hermes can also be used to predict the effect of mutations on protein-protein binding affinity. And it can be done in the same exact manner by using the predicted difference in log probabilities between the two amino acids. And in particular, we find that it seems to work best for mutations that occur at protein-protein interfaces. And I think this makes sense, and I think that it's because mutations that are occurring right at the interface are more likely to be affecting binding affinity through local effects. Whereas if you were to have a mutation like farther away from the interface, the way it would affect binding would likely involve sort of long-range chain of interactions, which Hermes would not be able to model. And so in the final application that I will share with you today, I used this ability of Hermes to model binding affinity differences at interfaces to tackle an important problem, or to try to tackle an important problem in immunology, which is to characterize T-cell receptor specificity. So let me start with some, again, biological background. So once again, we are in the immune system, specifically in the adaptive immune system, where one of the key players are T cells. And in particular here, we're talking about cytotoxic T cells. And each T cell carries on its surface a hypervariable receptor. It's mostly unique. That is called a T cell receptor, or TCR for short. And the job of these T cells is to use their TCR to scan peptides that are presented on the surface of cells and are essentially pieces of the proteins that were previously inside of the cell. And to put it simply, as you can see here, if a TCR binds to a peptide that is usually presented on top of an MHC molecule, if the TCR binds the peptide with sufficient affinity, it implies that the cell is pathogenic because it contains some pathogenic protein, and so this activates the T cell and ultimately results in the killing of the pathogenic cell. And sort of because of this ability to bind peptides and kill cells, it's also obviously important that TCRs don't bind to so-called healthy self peptides that belong to healthy proteins that are constantly sort of being presented by the cells. And so, because of this sort of selectivity that every TCR has to possess, a very important question in immunology is to figure out the specificity of a given TCR, meaning figure out which peptides a given TCR can bind to and elicit a T-cell response. This is important not only because it can answer sort of long-standing questions about how the immune system works. In fact, as it stands, we only have theories of exactly how specific TCRs even can be in order to provide immune coverage. But also, there are many translational applications that derive from can be aided by understanding TCR specificity, for example, T-cell therapies for cancer immunotherapies as well as cancer vaccines. And so for this problem, what we propose to do is to very simply use Hermes to predict amino acid probabilities on all sites of the peptide, as it is shown in the context of a specific TCR-MHC structure, and to then use these predicted probabilities to both score peptide sequences for their predicted affinity to the TCR, and to also use these probabilities to sample peptide sequences that are predicted to bind to the TCR and elicit T-cell activation, thereby basically mapping or at least sampling from the TCR specificity landscape. So we'll only talk about the sort of design portion of this work. So to design peptide sequences, we have two approaches. Both start from a known TCR-MHC structure that contains some wild-type peptide. And the first approach is based off of Hermes fixed, which simply involves sampling amino acids from the probability vectors that are predicted by Hermes. You just sample an amino acid at each site of the peptide, and thereby sort of construct site by site the peptide sequence. And doing this, you can actually sample a lot of peptides very quickly because you just need one forward pass of the model, or at least one per site. But the problem or the limitation of this is that it assumes that the structural environment is unchanged for every peptide that is sampled, so it basically ignores the space of peptide sequences that would potentially bind to the TCR, but with a different structural conformation than the wild types. And so to try and sample from this space, we then developed another pipeline that we call Hermes Relax, which is essentially an iterative algorithm that samples a peptide sequence like before and then mutates and relaxes the structure to contain the peptide sequence, and thus repeats this to optimize the peptide sequence, the predicted score of the peptide. And then we filtered the design peptides we're using a method that is very common in protein design these days, which is to use the predicted confidence of a structure prediction algorithm that is being inputted with the sample sequences. And specifically here we use a model that is a version of AlphaFold that is fine-tuned to work well on TCR-MHC structures. And so with using this pipeline, we design peptides for 3 TCR systems that target diverse wild-type peptides. And our experimental collaborator, Aja, measured T-cell activation as a result to exposure to these design peptides. And we were able to design many peptides that activated T cells across the 3 systems with varying degrees of success. And so to analyze this a bit, we found that many of the successful peptides were similar in sequence identity to the wild type, as you can see at the bottom, indicating that likely most of the peptides that we designed would bind the TCR with the same binding pattern as the wild types. But also, we can note that as you increase the sequence distance to the wild types, the design space of peptides increases a lot, as you can see in the top plot. And even at a distance of 5 amino acids away from the wild type, which is more than half of the peptide, we have a consistently high success rate of 20%, which is against the backdrop of about 10 million possible peptides that we could have sampled from. And so Hermes was definitely successful at navigating the space, and I think at the very least Hermes was able to identify amino acids on the wild types that were important for T cell activation and to propose substitutions that were neutral in terms of binding affinity. But that's not all, because in some cases we were able to also design peptides that elicited stronger T cell activation than the wild types. And in particular, for some of these cases, we believe this is driven by the presence of a glutamic acid at position 8 of the peptide, which we predict engages in a very strong polar bond, a salt bridge with an arginine on the TCR, which is an interaction that is not present in either of the wild types. And so I think this means that Hermes, the ability of Hermes is not limited to neutral mutations, but also to, it can identify mutations that improve interaction as well. And last, we were hoping that Hermes Relax would be better at identifying peptides that activate T cells while being very different from the wild types, because it had this option of sampling structural flexibility. Unfortunately, we did not observe this. As you can see in the bottom, the success rates are pretty equal. And I think I have some reasons to believe that our relaxed pipeline has some shortcomings, but I also think this might indicate that Hermes Fixed is a good enough model of these mutations. And I actually think I found some evidence indicating why Hermes Fixed might be a good model of, or at least a predictive model of neutral mutations in TCR-PMHC interactions, because I found this plot in a totally unrelated paper. And what this plot shows is that if you take all the known binding affinity measurements between TCR and PMHCs, binding affinity differences in TCR-PMHCs, you find that the neutral mutations, so mutations that preserve a binding affinity, also tend to be between amino acids of very similar sizes. Like, so there's a correspondence between size preservation and affinity preservation. But if you remember, is also correspondence that was found for Hermes Fixed. And interestingly, this size preservation is stronger than the preservation for other properties, and it is also strongest for TCR-PMHC mutations over other types of protein-protein interactions. And so I think, I speculate that perhaps this, similar bias that the system and the model have, whether it is for the right reasons or not, might make Hermes Fixed help make it a good predictor in this system. So in conclusion, today I've hopefully shown that this framework is successful for developing models across different areas of protein function prediction and design. And this framework hinges on three main design principles that I've thoroughly explored throughout my work, and I just want to conclude by discussing what I've learned about their strengths and weaknesses. So one of these principles is this residue-based locality, which throughout this work I've come to think is applicable often enough to be worth the simplification that it provides. And also I do think that this is a limitation that is very interpretable and known a priori, which I think it can be useful in practice, because in machine learning we often fall into the fallacy of believing that because our model has the capacity of learning something, that it will actually have learned it once it was trained, which is actually often not true, but it's very hard to probe whether that has happened or not. And so I think that if you are able to make a good model that works well within a specific context, and that context, then you have a model in your tool belt that you know how to use, and when to use it, and will be useful when you will need it. Which is a very engineering way of thinking about it, I think. Then another principle is all atom modeling, which I juxtaposed against modeling only protein backbone structure and amino acid sequence, which I didn't talk much about in this talk, but there is a bit of a sort of strain between these two approaches. And I think all-atom modeling definitely improved the predictions of our sidechain packer, because we had a first not all-atom prediction that was refined by an all-atom approach, and so I think all-atom modeling definitely paid off there. But also, this approach is what is at the basis of these size-dependent preference that is in Hermes, in particular in Hermes fixed, which again, through my analysis, I found to be a double-edged sword. That sometimes this bias is a hindrance because it's limiting in the type of things you can predict, but some other times it's sort of a bias that is actually matches a bias that is present in your system, which I think is quite interesting. And last we have rotational equivariance, which I didn't say much about because I wanted this talk to be more high level and a bit more about the biology, but what I'll say is equivariance was it was very hard to learn the theory of it and to learn how to apply it to neural networks. And in fact, I found through talking to people, I found that many people in this community just try to avoid using it and learning it because there's sort of a initial cost in learning it that people are afraid it's not going to pay off. And I do think that it is worth the pain, the data efficiency that it provides. But in particular, I think it is worth in the context of this framework because many folks have found that some of these types of models tend to be very very slow and very difficult to optimize, and instead this particular framework is simple enough that those problems don't exist, but you still have this nice equivariance. And so I do think that this framework has this potential is still can still be developed. So that concludes the part of my talk that is more technical, and now we'll spend a few minutes to try not to cry and not look at people in the eyes. I thank the people that have made my that have helped me throughout my PhD in one way or another. First of all, thank you, Armita, for answering my cold email a few years ago. You had sent like an email trying to get students, so I wasn't that cold. But thank you for welcoming me, for teaching me the ways of physics, the ways of equivariance, and I think Armita and I share a very scattered curiosity for understanding how things work, like whatever they may be, and biology is a very fun playground for fun problems, and I think I have benefited a lot from this relationship and for having sort of a like-minded person in that regard. So thank you a lot for that. I want to thank Mike for his mentorship in the first years of my PhD. I tend to be quite impatient and hot-headed, and Mike is the opposite, and that probably has caused tensions between us that maybe he didn't even tell me, but I'm sure they have, because at some points later I found that some of the things I didn't understand about what he was doing or how he was doing things actually were right and I was wrong all along. And thank you, Mike, for teaching me in my first years. Thank you, Zach Montague, for always being an element of support in the group. Thank you, William, for just being a lot of fun to work with. Like, I just enjoyed it, so thank you. Similar for Kevin, thank you for always being down to brainstorm things and to teach me the ways of math, teaching me things that I wish I was able to learn by myself, but you were always there to teach them to me, so thank you. Now I have Claude, but you're still better. You're still better. Thank you, Zach Jones, for being an excited collaborator and for always being available to teach me the ways of biology. Thank you, Will DeWitt, for hosting me in the last few months and for recruiting me in the academic adventure that Armita mentioned at the beginning of this talk. And This is just thank you to my friends who have made these 5 years fun in Seattle, who have shared my hobbies and showed me theirs, and have eaten all the things that I've tried to cook for them over the years. I think they've enjoyed them, but sometimes I've let them down, and it's fun. In particular, Artie, my first friend here, my fellow Italian, is probably the most thoughtful person I know, and I just thank him for that every day. Lancelot, who is always down to clown, and I think I've showed that salmon video to so many people, for being my emotional support person throughout these years. Kevin, for always being down for fun Teletubbies adventures. Shout out to my Italian friends over here in the back, dressed nicely. Why are you laughing? They've cherished my friendship throughout these 10 years that I've been abroad, and even came all the way here to be with me today, and I'm super grateful for that. Thank you to my partner, my beautiful partner in life and in crime, Ali, who is always supportive of all the things I get down to, and is super patient with me, and in these years, in these days especially, that have been super hectic. She's been super helpful, and most importantly, as you can see here, she's the creative muse of my life. She adds color and spice to my everyday life. I love you very much. Thank you. And I have it written, so it's easy. And thank you to my family for supporting me and supporting my career, which I don't even know what it is yet, while enduring my absence, for always being available to use their knowledge of me to help me through life and through these years. They say Italian men are mama's boys, and usually that's an insult, but it's true. I mean, at least for me. So, yeah, it's been hard to be apart, and it's the biggest cost I've paid, but and it's also been great to finally share my growing love for biology with my sister, who grew up without me for most of her life, and now she likes biology too, and it's been a lot of fun. So it's not a thank you, it's just nice. And with that, I'll take questions, and thank you.